Our chronological age is how many times we orbit the sun. Every year we get to add one more, right? Doesn't matter. It's relentless. Your biological age is different. Your biological age is a representation of your functionality, and it is not necessarily directly linked to your chronological age.
This is the Transformational Life Consulting Podcast with Deepa Grandin, MD, where Christians like you get the practical support and tools you need to build your spiritual, mental, and physical well-being, and to guide you on your journey to health and wholeness based on evidence-based science and God's Word. I am your host, Dr. Deepa Grandin. The title of today's podcast is Understanding Causes for Premature Aging of Your Heart and Blood Vessels and the Role of Plasminogen Activator Inhibitor 1, or PAI-1. In one of the longevity webinars conducted a year ago, I went into the details of the difference between biological age and chronological age and the importance of knowing the difference between the two. This episode is accessible for TLC members in the archived videos section. Please listen to it if you haven't already. For those listeners who are not members, some basic definitions will be revisited in today's podcast. For the most part, for today, we're going to focus on factors that cause aging of the heart and blood vessels, specifically looking at the role of PAI-1. So what exactly is PAI-1? So PAI-1 is a protein that has been identified as a critical contributor to aging at a cellular level. And so this is, going to be an important topic to talk about because we know that your chronological age, that's the age or how long you've been on this planet, versus your biological age, which is really the age of your cells based on how well or what type of choices you make on a day-to-day basis, is determined by so many different factors. And PAI-1 is just one of the many proteins that do play a role in how our cells age. And what research has shown is when you have really high levels, it can predict the development of aging-related disorders such as type 2 diabetes, heart disease, hypertension, and time to heart failure. Today, we are honored to have Dr. Douglas Vaughan, who is a renowned expert in this field. He is going to help shed some light on this important topic, and we are so grateful for his time and expertise. Dr. Vaughan is the Irving S. Cutter Professor and the Chair of the Department of Medicine at the Northwestern University Feinberg School of Medicine in Chicago, Illinois. He received his MD from the University of Texas Southwestern Medical School in Dallas and completed his internship and residency in internal medicine at Parkland Memorial Hospital Veteran Administration Medical Center, also in Dallas. He subsequently undertook a fellowship in cardiology at Brigham Women's Hospital and Harvard Medical School in Boston, Massachusetts. He is a fellow of the American College of Cardiology and has been elected to membership in the American Society for Clinical Investigation and the Association of American Physicians. As a principal clinical investigator, Dr. Vaughan's work ranges from the bench to the bedside. His efforts and findings in vascular biology include basic investigations into the regulation of gene expression, genetic models of disease, mechanistic studies in humans, and clinical trials. His primary research interests are in the mammalian plasminogen activator system and the role it plays in cardiovascular disease and physiological aging. Thank you, Dr. Vaughan, for being here with us on this podcast. The audience and I would love to get to know you a little bit better. So if you don't mind sharing with us a little bit about your journey, what led you to cardiology and research in your area of specialty?
Well, first, thank you very much, Deepa, for having me on your program today. I'm delighted to be here and to have this conversation with you. But yeah, You detailed my biography pretty well, but there are a couple points I'd like to make. For the last 25 years, I've had major leadership roles in medicine. First as chief of cardiology at Vanderbilt for a decade. And then in 2008, I moved here to Chicago, where I've been chair of medicine for the last 15 years. And, over that period of time, I learned a lot. We won't talk about all those things today, but one of the things that I became more and more aware of is the fact that people age at different rates. And I'm talking about just observing patients. You can walk through the hospital and you can see individuals that appear much older than their stated age. And you occasionally see people on the other end of the spectrum that appear to be younger than their chronological age. And I think all your viewers and all your listeners are probably aware of that in their everyday life. They know they have friends or family that appear to be protected from aging, others not so much. But that's kind of told me, and it's sort of informed me and catalyzed my interest in exploring what causes these differences. Mm-hmm. And the fact that there are differences suggests to me and others in the field that maybe we can do something about it.
Yeah, for sure. So that's so true. I mean, I don't know. I'm sure you're familiar with Dr. Michael Roizen.
Sure.
He's the chief wellness officer at Cleveland Clinic. And I love his book, The Great Age Reboot.
Yeah.
And the way he explains it is like, think of the choices you make on a day-to-day basis, like you're putting money in a bank.
Yeah.
Because that way you're extending years of life, and not just extending life, but quality of life. So I think the work you're doing is amazing, and it's so important.
Well, my research program is multidisciplinary. We go from the bench to the bedside. It's a terribly fun and exciting way to live and work and think. My research gradually migrated from cardiovascular disease in a very specific sense, more broadly into aging. And that evolved for a variety of different reasons. One related to the fact that the single component that we're most focused on in my laboratory, PAI1 or PI1 for short, turns out to be one of the molecular fingerprints of senescence. That was discovered in the mid-90s. And that started to flip a switch in my mind to think about PI1 as a contributor to aging, not just as a marker of aging. And then we explored that at a number of different levels in the laboratory. And we found that mice that express too much PI1 develop phenotypic changes or manifest changes that look like aging. Mm-hmm. Conversely, mice that have less PI1 due to genetic manipulations or by drugs seem to be protected from a variety of forms of aging. Mm-hmm. And that really catalyzed my dedicated effort to go study members of an old order Amish community that live relatively near to Chicago. They're in northeastern Indiana, in and around Adams County. They settled there from Switzerland in the mid part of the 19th century, but this is a single community in the world that harbors a null mutation in the gene that codes for PI1. Wow. And out of 20,000 members of the community, there are probably 3,000 carriers of this genetic variant. They don't know it. They're not genotyped at birth. They don't feel anything from it. But several years ago, we went over there and actually studied nearly 200 members of the community, and we found that the carriers of this genetic variant are protected from aging in all kinds of ways. Their telomeres are longer, their fasting insulin levels are lower, they don't get diabetes, their cardiovascular system retains its juvenile characteristics, and they actually live longer than their unaffected kindred. So I think it's a single example of a genetic variant in the world right now that protects a bit against biological aging in a variety of different ways. So you can imagine how lucky I feel that the single community in the world, maybe in the whole galaxy, that has a PI1 variant.
Yeah.
Highly represented in the population, is only a 4-hour drive from Chicago. So I operate a satellite laboratory over there now. We continue to expand our studies of that population, try to understand how this provides some protection against aging in the human species.
I think it would be really interesting also to evaluate the blue zone populations to see if they have something similar.
Yeah, I think the blue zones are really, really interesting. I think most of those represent environmental effects on aging, whether it's climate, diet, society, culture, all those sorts of things. But they tend to be very, very healthy people. They tend to have very good diets. And the link with Pi1 might be highly relevant there, because it turns out that the most important determinant of the levels of Pi1 in your blood, in your body, is your body mass index.
That's right.
So if you're lean and you're fit, you're going to have a low level of Pi1. So that's not going to be a contributor to your aging. Now, there will be other things, but they're likely, that's probably one component of the many factors that protect people in blue zones, as opposed to a single gene variant.
Okay, that's good to know.
Mm-hmm.
So, Dr. Vaughan, you're also the director of the Plasminogen longevity. I don't know if I'm pronouncing that right.
Plasminogen. The C is silent.
Okay, Plasminogen Longevity Institute. Can you tell us a little bit about that institute and why was it established?
Yeah, it's a relatively new creation. Our dean here at Northwestern convenes a large group of faculty at regular intervals, and about 6 years ago, we come up with ideas or areas that we want to focus on as a larger group. And at one of those meetings a few years ago, what bubbled up was interest in artificial intelligence and healthy aging. So that sort of motivated the school to begin a program to raise money to support those initiatives. And actually, they found a very generous donor and his family, John Podesnak, who are interested in aging and longevity. And they gave us a very large gift. And the Podesnaks are interested in aging from a little bit different perspective than most might think. They're interested in us doing things for people that are disadvantaged with regard to aging because of chronic conditions or circumstances, like, for instance, chronic HIV infections.
Sure.
People with chronic HIV appear to age more rapidly than others. The Podesnaks want us to do something about it. That's why they gave us the money. But I find that very empowering and motivating to think about. We want to do something to help people that are on the fast slope, rapid slope of aging, as opposed to protected from aging. And so that's what we're motivated to do. Hopefully, we'll be able to find some things that actually impact upon people that are disadvantaged in this space. And certainly, there are a lot of people like that in the world.
Yeah, I think that's such a great cause. And as far as, because we have a mixed audience of people who are both in the medical field, but also the general public, and they may not all be familiar with what is biological age and how is that different from chronological age. Do you mind shedding some light on that difference?
Yeah, this is something we talk about a lot, and I think this is a topic, this is a concept that's become clearer over the last decade or so. So our chronological age is how many times we orbit the sun. Every year we get to add one more.
Yeah.
Right? Doesn't matter. It's relentless. Your biological age is different. Your biological age is a representation of your functionality, and it is not necessarily directly linked to your chronological age. And this is important because, we're at a really unique moment in human history. There's a convergence that's happened relatively recently, where we've really largely we've made huge progress in demystifying the biology of aging. I think we were starting to understand that pretty well. At the same time, we have the capacity to measure biological age with a precision and a validity that never existed before. And those two things converging now in 2024 gives us the idea that perhaps we can do something about aging. We understand the causes and we can measure it. So can we actually do something about it? And that's the moment we're living in right now. And that's sort of what our institute is about. Our institute is comprised of 5 different centers. We have a center for basic and translational science and aging. We have a center for population science and aging. We have a center for HIV and aging, because that's a particular interest of the Podestacks. We have a center for nanoscience and aging. That's a rapidly developing field. And the center that's most externalized, and maybe the one that people are most aware of, is what I call our human longevity laboratory. And that's where we bring people in and we measure their biological age in multiple dimensions. We put them through a series of interesting tests that test their function and structure in a variety of different systems. We also do molecular profiling that gives us other information about biological age. And we also, some of the tests that we do are analyzed using artificial intelligence, and that gives us a different version of your biological age. And so we integrate all those things at the same time. It takes about 2.5 hours for a person to go through all the testing, but at the end, we give them a report card, and we tell them where they stand, at least at this moment in time.
That's amazing. And are you inviting people from the general public to come and do this assessment?
Yeah, we are. And, that's sort of one of the beauties of this gift from John Podestack and his family. We have some resources to be able to bring people in and study them. So right now we just really opened the laboratory in the last 6 months. So we're bringing in 2 people a day.
Okay.
And we put them through these studies, and we're looking at a cross-sectional view of Chicago or whatever. We want people of all different ages, all different races, all different ethnic backgrounds, all different socioeconomic groups. We're trying to get a handle on what aging looks like in a population. Within the next 6 to 8 months, we will pivot into doing interventional trials, randomized trials, where we look at specific interventions to see if we can't slow the pace of aging in individuals. And, you can imagine the kind of things that we're talking about testing. It might be as simple things as dietary changes and intermittent fasting or exercise or stress reduction, but also novel therapeutic ways of doing it. Maybe drugs like Ozempic slow down aging. I don't know.
Okay.
But we'll have the capacity, and that's really our pragmatic goal of the Human Longevity Laboratory, is to do studies, relatively small scale studies, that allow us to get a vision and develop an appreciation for interventions that might actually impact on aging in the human species.
Yeah, that's really fascinating. So if, say, somebody's interested in participating in this analysis, what's the best way for them to reach out and get connected?
We have a site on our website. We have the opportunity for people to give their information and get their name on the list to potentially come in and do this.
Okay, great. So maybe you can share that website with me, and then I can put that in the show notes, and people who are interested can reach out. So thanks for sharing that with us.
We've had reporters here from NPR and the Today Show over the last 3 months, and then they did some, and both those broadcasts were well received. I think we got several thousand inquiries.
Oh, wow. That's amazing. I can totally see that, though.
Yeah.
It's a very fascinating topic. So at the very start of this dialogue, Dr. Vaughan, you had mentioned that it's very obvious to see some individuals who are older than their stated age, and then others who look younger than their stated age. What do you think is causing that difference? Like, why do some people age faster than others?
That's a really fundamentally important question. And I think there's probably several different things that drive accelerated aging. Many people think that chronic inflammation drives accelerated aging, and that's present in a variety of different ways, and we see that. I think there are also metabolic forces that accelerate aging at the cellular level, and then that gets manifested more systemically. But we're also finding that there are, other things in the world that impact upon age, and it all depends upon how you measure it. But my colleague, Dr. Li Feng Hao here, studies population science and aging, and she recently had a very interesting paper published that showed that living close to a green space slows your aging. That probably shouldn't be too much of a surprise to your listeners. So your environment, the air you breathe, the food you eat, the chronic conditions that you have. Well, let's say you have chronic kidney disease. People with chronic kidney disease age very rapidly. There are factors in the blood. I don't know. I don't even think we know what those factors are exactly that drive accelerated aging in that population. If you're a 25-year-old on dialysis, you have the same cardiovascular risk as a 75-year-old.
Wow.
And it's really important though, because, the most important risk factor for every disease that we deal with in the adult population is age.
Yeah.
Age is the most important risk factor for heart disease. Age is the most important risk factor for cancer. Age is the most important risk factor for dementia or Alzheimer's disease. Age is the most important risk factor for diabetes or metabolic disorders. Get the trend there?
Yeah.
Something about aging that makes us susceptible to disease. We lose some of our regenerative or reparative potential as we age, and there are conditions and circumstances that make some people age more rapidly than others. And then, occasionally we see patients here at Northwestern that have genetic versions of accelerated aging, people with short telomere syndromes and things like that. Those are not that uncommon, but there are obviously other factors today. But the fun thing about it, and the amazing thing about it, is how precisely we can measure biological age these days. And probably the gold standard for measuring biological age right now involves looking at the pattern of chemical modification of your DNA or methylation. Your DNA, it's called your epigenetic age.
Right.
There are a variety of different biological clocks out there. Steve Horvath at UCLA is really the pioneer in that area, and has developed clocks that are informative not just for humans but for hundreds of mammalian species.
Wow.
So it's a true phenomenon. The pattern of your methylation does determine your biological age in some ways, and methylation is malleable. It's changeable. You can change your methylation just by diet and exercise in a relatively short term. So that gives us hope that perhaps we can actually do something about biological aging in the not too distant future.
Yeah, because I've heard it being said you don't have to go to MIT to become a genetic engineer. You can do that in your kitchen based on what you eat and,
Well, you're not changing the sequence of your DNA per se, but you are changing these methylated groups and other adducts that stick to your DNA and change the functionality of your genes.
Yeah. So you did mention the DNA methylation measurements.
Yeah.
So what, in your opinion, because you've been able to do a lot of hands-on research as to what's the best way to calculate biological age, can you share with us what your takeaway is?
Well, the more I look at it, the more I think that that you need to have a multidimensional view of this. I don't think it's a single perfect measure of biological age, although epigenetic age is a contender for that. We do a variety of things in our laboratory that register biological age. For example, we do a simple thing. We just take a photograph of your retina, and we collaborate with a group in New Zealand called Toku Eyes, who have used machine learning to analyze retinal images from hundreds of thousands of individuals. And now they have an AI-based program. They can look at the picture of your retina, and they can tell you your biological age in about a nanosecond. It's amazing.
And does that correlate with other forms of calculation?
Highly.
That's amazing.
Yeah. We work with a group in Brazil that has used machine learning to analyze millions of 12-lead electrocardiograms.
Mm-hmm.
Well, most of your listeners have had an electrocardiogram done in their life. I'm a cardiologist. I can read an electrocardiogram, but this AI-based tool can look at an electrocardiogram and tell you your cardiovascular age in a nanosecond.
Wow.
So there are all these kinds of tools that are developing now. So that's what we do in our laboratory. And we do epigenetic age, but we also do retinal age. We do ECG age. We do other measures of biological age. We think it's an aggregate that is most informative, and which ones are going to change. I mean, if your epigenetic age gets younger, but your cardiovascular age doesn't change, have you really helped anybody?
Mm-hmm.
Nobody's addressed this question yet. Yeah. These are the kind of questions that we want to see if we can look at, because at the end of the day, aging is about what happens to your structure and function of our systems in our body.
Right.
That's all. If your epigenetic age is 1, but you have the cardiovascular system of a 90-year-old, I don't think it's going to help you too much.
Yeah, true. So, I mean, I think you've made a very important point about the multi-dimensional approach of looking at biological age. But there's also mention in the work that you do about multi-dimensional phenotyping. Mm-hmm. And so how does that measure biological age in your participants?
Yeah, so we're really interested in the physiological changes that occur as a function of age. So we do a variety of measures. We could put our participants through a lot of different tests of systems and functions that we know change with age. For example, we test their hearing. Mm-hmm. Most people lose some of their hearing as they age. We test their sense of smell. Yeah. That tends to deteriorate as a function of age. We measure a variety of different things in the cardiovascular system that we know change as a function of age. For example, the speed with which blood flows through your aorta accelerates as your aorta gets stiffer or as you age. So we measure your pulse wave velocity. We measure your heart rate variability. A lot of people wear rings that measure that, but your heart rate variability compresses as a function of age. That's another kind of, it's not a biological age per se, but certainly it's a factor of, or a function that changes as we age. We measure pulmonary function. Mm-hmm. We measure we do a series of tests around musculoskeletal function, because almost everybody as they age realizes that they've got a joint or two that are bothering them, or they slow down, or their walking speed slows down. So we actually collect that sort of information and actually analyze gait using AI as well. So all those kinds of things, we try to touch on every system that we can in non-invasively and safely. Yeah. We don't capture anything specifically about the gastrointestinal system, but maybe we will in the not too distant future. But for the time, that's part of our sort of universal view of aging, that we want to capture data from as many different systems as we can. And as we test interventions that we think might impact upon biological age, we would hope that would register in one or more of these physiological measures as well.
Okay. Yeah. Because there are so many, I mean, like, it's still an evolving field, and it's so hard to pinpoint, like you said, one specific thing. They all kind of contribute in a collective way.
Yeah, and and there are really interesting papers that have come out recently looking at epigenetic age, or the pattern of proteins in different organs in the body, and actually in one individual, organs age at different rates.
Interesting.
Yeah, so we have to deal with that variable as well.
Yeah, and you've mentioned the AI factor multiple times. Can you tell us a little bit more about how you're using AI in your research?
Yeah, so I mentioned two specific collaborations that use AI to create a calculation of biological age. We're also collaborating with a group out of Oxford, England called Ultromics. They can analyze an echocardiogram with a single 4-chamber view, and we do a limited echocardiogram on our participants that go through the Human Longevity Lab, and they can tell you with a AI program that they have whether or not there's diastolic dysfunction or impaired relaxation of the heart. That's an aging-related phenomenon. Okay. So that's another example. I have a colleague here at Northwestern, James Cotton, at the Shirley Ryan Ability Lab, and he's developed an AI tool where he takes a simple video of our participants while they're walking using his iPhone. He puts a couple sensors on their lower extremities, but then he can go back and analyze it for gait speed and propulsive velocity and balance in ways that are amazing. And that's another component of AI that we use. But there will be more and more AI-based measures of biological age. I was at a biological aging meeting in Barcelona last week, and all the leaders in the world were there. And I heard a story about a biological age measure created by a group in Beijing that uses a thermal photograph of the face.
Wow.
And that can register biological age, at least with the tool that they have using AI. And that's pretty interesting. But there will be others. Stay tuned to that space because there are a lot of work there. Yeah.
Yeah, I wonder if that has anything to do with the mitochondria and how.
Probably, yeah, and respiratory efficiency and function, as well as simple things as blood flow.
Yeah. Fascinating. Yeah. So in your experience in research, what interventions can alter the aging trajectory for individuals facing medical conditions or other challenging circumstances?
Well, this is going to be boring. This is the one thing that works right now: diet and exercise. Right. That's proven to be true.
Mind-blowing.
Yeah, I know. It's just amazing breakthrough, right? But that's hard. And are there other things that can work? One of the first things that we're going to study in our laboratory is stress reduction.
Exactly.
A lot of people think that stress ages them. I think that's probably true. We can measure that though, and over time we intend to bring in individuals from our first responders here in Chicago, see what their biological age is now, how that kind of stress is impacting their life. We hope eventually to study veterans that have been through combat, see how that affects their biological age. So stress reduction, there are lots of people out there in the world that think drugs like metformin can slow down aging. I'm skeptical. Let's do some rigorous studies and measure age, a variety of different dimensions, and see if that really works. It might. I'm open-minded about it, but we don't really have the kind of multidimensional studies, randomized, placebo-controlled, to say whether or not that really does work. But it might in some people. I don't know.
Yeah. So that's interesting you said that, because just recently I was listening to a book that was talking about a research study that looked at people who intentionally incorporated stress reduction interventions. And it did show from a biological level that they were able to change some of those epigenetic aging factors. And the other thing about the metformin, there was a recent study in asthmatics, because we know at a cellular level, asthma is very much an inflammatory disease. And metformin was able to even alter their lung function. And this was like a incidental finding, if you will. So individuals who were on metformin and, and they did their lung function tests, they tended to have better preservation of lung volume and just overall how the asthma panned out over time. So it would be interesting to see from a longevity perspective?
Well, there are lots of potential candidates that have been identified in experimental studies in a variety of different species, everything from worms to mice. There are a lot of candidates. Metformin's one of them. Rapamycin, certainly popular among the basic scientists in the field of aging. There are other dietary supplements, taurine, for example, which is mostly in shellfish, but that appears to be, at least in populations studies, suggests that eating shellfish might have a protective effect on aging. You can imagine there are so many different things that are candidates.
Yeah.
It's hard to prioritize, which one. Yeah. We're really interested in finding interventions that are safe, um, inexpensive, equitable, and that can be delivered to all kinds of people without breaking the bank.
Yeah.
My big dream right now is to create a global network of human longevity laboratories like we have here in Chicago. And that laboratory maybe in Abu Dhabi or in Asia, in Europe, in South America, and all of us do the same studies with the same equipment and the same measures and test interventions together and see how they impact upon people in different climates and different societies and with different ethnic backgrounds and different diets, and see if we can find some truth about what really impacts upon the human condition and share that information, with the world.
Yep. Yeah, I think that would be amazing because we
Does that sound too grandiose?
No, I think that's actually quite possible. And having that kind of collaboration would be very necessary. So we're not just looking at one pool of population, but multiple different, and how it pans out across the board of different ethnic groups.
I think that's absolutely necessary.
Yeah, I agree. Yeah. So your main area of research interest is the Plasminogen activator system. What role does this system play in cardiovascular disease and aging?
Yeah, I developed my interest in my addiction to studying the plasminogen activator system when I was a trainee and a cardiology fellow. That was the time that TPA was developed and used as a clot dissolving drug, and so I became fascinated with that clot dissolving system in the body. And PI1, the protein that I work on, regulates our clot dissolving system. So excess PI1 is a factor that increases the risk of clotting. We found out now over the years that it's involved with many other things. It's involved in blood pressure regulation. It's involved in metabolism, both carbohydrate and lipid metabolism. We know that PI1 regulates the activation or clearance of a number of different peptide hormones in our blood. For example, when you make a new memory in your brain, you have to make a hormone called BDNF. BDNF is generated in a precursor state called pro-BDNF. Well, it's actually activated by the action of TPA, that clot dissolving drug. Well, it's not just a clot dissolving protein. And it turns so PI1 blocks the proteolytic activation of BDNF. So if you have too much PI1 in your brain, you have a hard time making new memories. PI1 influences a number of different factors that are involved with lipid metabolism. We found that PI1 regulates the proteolytic degradation of PCSK9, which is a critically important factor in our cholesterol metabolism.
Right.
We even give PCSK9 inhibitors to patients with cardiovascular disease. So broadly, it's involved in a number of different systems that impact on disease and aging-related diseases.
Yeah, so that's interesting because we also know that exercise increases BDNF, or the brain-derived neurotrophic factor. So that would be a good way to overcome the PI1s.
One more benefit of low exercise and a lean body mass.
Yeah, I agree. So what does the thrombotic cardiovascular, or I should say, why does the thrombotic cardiovascular disease increase with age?
That's a very important question. And we certainly see that, as I mentioned a few minutes ago, age is the most important risk factor for cardiovascular disease, things like heart attack and stroke. And the proximate cause of those clinical events is usually a clot forming in an artery. There are lots of ways we protect ourselves against clots forming in our arteries. We have a system of endogenous anticoagulants, if you will, to protect us from clotting, protein C, antithrombin 3, et cetera. Luckily, we have those around, or our blood would just turn to a gel.
Yeah.
We have a system of short-lived endothelial-derived inhibitors of platelet function, like prostacyclin and nitric oxide. Those systems prevent clots from forming. And then Mother Nature's been very concerned about it. She gave us a backup system, a clot dissolving system that includes the activity of our fibrinolytic proteins like TPA and plasminogen. So all those work together to keep us from forming clots in our blood vessels or to clear them if they form. And those all change as a function of age. And certainly the ability of our endothelial cells to make nitric oxide deteriorates as a function of age. So we lose that platelet inhibitory function. On the fibrinolytic side, we tend to see that Pi1 levels increase with age, and certainly Pi1 increases in cells that have adopted a senescent program. So that's one of the factors that contributes to increased risk of clotting as a function of age.
Yeah, and I think an important point I would like to highlight is when we're talking about aging, it's at the cellular level, not necessarily a chronological age. And obesity, as we know, is a great cellular ager.
Yeah.
And so that's another reason, people really need to be taking to heart at a personal level, how do I, take it most seriously as far as losing weight and making better lifestyle choices. And I think now that you're talking about the Pi1 and how, as the cells age, that increases exponentially. So that's a good point to make.
Well put.
Yeah. So is there anything else, Dr. Vaughan, that you would like to share with us?
We've covered a lot of topics. I hopefully I've shared and made it clear how excited I am to be in this space right now. That this convergence that we're witnessing between understanding the biology of aging and being able to measure biological age in humans, this gives us great opportunities and optimism that we can do something about it. There will be all kinds of investments and opportunities to impact on age. There's a huge amount of money flowing into aging. There are foundations that are supporting aging research, like the Evolution Fund, the Impetus Funds. I think those are great contributors to the space beyond the typical sort of governmental research support. Also, there are amazing and massive investments being made in aging by companies like Google and Amazon.
Yeah.
Hundreds of millions of dollars going into this. So, stay tuned. There will be pretty interesting things on the horizon, maybe sooner than we think. But I tell my students, I tell my residents, you really need to know about aging because you will be prescribing drugs that slow down aging during your career. And if you slow down aging, you'll be able to push back the onset of disease a little bit in your patients and give them a longer health span. And that's really what we're focused on and what we're trying to deliver. We're not necessarily trying to find a way to get people to live to be 150. We're trying to find a way to give people the chance to enjoy their life a little bit longer.
Exactly.
And do the things that they want to do, whether it's work or play golf or whatever they want to do. and that involves extending the health span. And that's what our priority is here.
Yeah, that's amazing. It's extremely exciting. And so anyway, thank you so much for being here and sharing your knowledge with us. And we look forward to collaborating with you in the future.
Thank you very much. It's been delightful talking to you and about this really exciting topic.